Chapter 23. So you can see we're kind of jumping around. We were going chapter 12, 13, 14, 15. This week we went 16, 17, and then we jumped to 20, chapter 20. That was yesterday, and now we're chapter 23, lipids today. So we are going to talk about what makes something a lipid, okay? And what is lipid is another word for what? What do we call lipids generally, usually?
Fats.
Yep, call them fats, okay? And we know that we need fats in our diet. Sometimes I think fats get a bad rap. Like, oh, don't eat too much fat, okay? You need fat for every single membrane of every single cell. And how many cells are you composed of? I mean, just billions, right? So fats are essential. They're really, really important. You cannot cut out on fats. It's just you don't need too much of the wrong kind of fat, okay? So does anybody know an example of a bad fat? You know, we have like these bad fats and good fats.
Trans fats.
Trans fats are bad fats. Okay, we're actually going to mention those in a little bit. Okay, why are they bad fats? Well, they've been associated with like heart attacks and heart disease and things like that. So that's one reason people are really iffy about trans fats. Okay, what kind of foods have trans fats?
Processed foods.
Yes, baked goods. Okay, this made me sad. I was looking specifically into this, but like refrigerated biscuits, okay, like the biscuit dough, like you just like take them out and pop them in the oven. I love to make homemade biscuits, don't get me wrong, but they are a labor of love. So occasionally I will buy canned biscuits, but those have trans fat in them. So margarine, anybody use margarine? We grew up, my mom would use margarine at our house, but I'm like, Mom, why'd you do that? Okay, margarine can be high in trans fat. So I'm a butter fan, okay, straight butter. Butter is actually a good fat for you, just not too much of it, okay? It's just with anything in our diets, we know everything is good, just in moderation, right? And so good fats, what are some examples of good fats? Good lipids.
Saturated fats?
Saturated fats are good.
And unsaturated, they're
We
okay.
need both. We need saturated and unsaturated, that's right. And we're actually going to define that in just a second. Okay, but I think like avocados have really good fats in them. Coconut oil is one that's just like become extremely popular, I feel like, in the past decade. So we've become aware of those as we've learned more and more and more about fats just from our diets and what we should and shouldn't eat. But what makes something a fat structurally, like the molecule? Well, I'll tell you this, fats can have all different kinds of structures. So it's not actually their structure. We have been so used to saying, okay, if you see this particular structure on a molecule, that makes it an alcohol or a carboxylic acid. And with fats, we throw all that out the window, okay? Lipids, what makes them a lipid, okay, is what it's soluble in, okay? Lipids are classified based on their solubility, not their structure. You can have widely different molecules with widely different structures. They could both be lipids, but what makes them a lipid is their hydrophobic portions, okay? They're mostly non-polar, or I'll say hydrophobic in character. And that makes them soluble in non-polar or organic solvents, okay? But they are naturally occurring molecules. You can have them from plants or animals, soluble in non-polar organic solvents. So largely their structures are non-polar hydrophobic, all right? We also have this term fatty acids, okay? And this is a particular type of lipid. You're just gonna hear me say fatty acid a whole lot today and moving forward. But these are lipids that contain long unbranched hydrocarbon chain carboxylic acids, okay? So we'll look at what that exactly means. Imagine that in your head. One portion, it's a long chain. One end has a carboxylic head and then just a long hydrocarbon tail. And that's where this hydrocarbon chain, this is the nonpolar part, right? And we've already discussed the effects of short nonpolar chains versus long when we looked at the solubilities of alcohols and amines and other molecules. All right. Now, when we bind these together, these fatty acids, a lot of times we'll bind them with glycerol. What you end up seeing is some kind of ester group or amide group. Okay, and I will be sure to point that out. For example, you can actually see right here, if you look at this, here's going to be, this part is going to be the carboxylic acid, and then this oxygen right here, I'll show you in a minute, this is actually going to be the ester bond, okay, on all these. But we'll look at a different example. It's kind of hard to see it from the condensed writing right there. Okay, so I wanted to show you this graph because there's all different kinds of lipids. So we are going to write out the biological significance of each of these. So write these down. This will probably be on your exam. Waxes. Why are waxes important? Okay, waxes are secreted by our skin. Anybody, your skin on your face especially gets oily. Okay, why do we use things like face wash? Because our skin is secreting. So these have protective significance or functions. Protective functions, coatings for the skin. Okay, one example, it's called sebum. Okay, our skin secretes this from our sebaceous glands. And some people just have especially oily skin. That's the way you might hear it described. Okay, that's why we use things like face washes. Triacylglycerols, okay? This one, the main significance is it's fat storage. Okay, so it's the major storage molecule if we're gonna store fats long term. But we use fat storage for biochemical energy production, okay? If somebody has a diet that's low in carbohydrates, or if you starve yourself of carbohydrates, your body will begin to use fatty acids for energy. Okay, we have a whole process and pathway for that. We're going to look at that. But triacylglycerols are the main type of lipid we use for fat storage. And so therefore, when we break those down, we get a lot of energy from those. Okay, glycerophospholipids. These are both triesters of glycerol. So structurally, they're kind of similar. But there is a difference with glycerophospholipids. They have this, y'all remember what a phosphate group is? Okay, this is the PO3 minus 2. Or sometimes you might also see PO4 with a minus 3. There can be both, PO4 with a minus 3. These are both examples of phosphate groups. But why are glycerophospholipids important? These, I'm going to draw an arrow and go up. These are the main lipid in cell membranes. So what do? They regulate, they control the flow of molecules into and out of the cell. Okay, so they regulate flow of molecules in and out of cells. Okay, they create the bilayer that is our cell membrane. Okay, so we've got waxes, triacylglycerols, glycerophospholipids. Now we're going to jump over to, this is a weird word, sphingolipids. There's two types of sphingolipids. We have sphingomyelins. Okay, these are the lipids that are abundant in nerve cell membranes. Okay, so they are abundant. So our nerve cells, we know, are a special type of cell, and they are constantly sending electrical signals to our brains and all over our bodies. They're, we have pain receptors there, and these they help protect those nerve cells. They kind of coat around them and protect them from damage. Okay, that's sphingomyelins. Okay, glycolipids. This is important for intracellular messaging. So a lot of times glycolipids sit on the surface of cell membranes. Or they have some kind of chain that sticks out that receives messages and sends messages. So they are intracellular messengers. Okay, they sit on the cell surface. Okay, so that's glycolipids. Steroids, we're familiar with steroids. One important type of steroid, I'll go up. Okay, cholesterol. Everybody's always concerned about cholesterol, especially the older you get. And then sex hormones. Okay, estrogen, testosterone, and there are others. These are steroids by structure, okay, but they're all lipids. And then the last one, eicosanoids is how you say that. These are specialized local, okay, so they don't send messages all over the body. They act more locally. cell messengers. So one example of eicosanoids, have y'all heard of prostaglandins? Okay, for example, if you get some sort of bug bite or a cut, and you have some sort of an inflammatory response, okay, especially with like a bug bite or a cut, your skin gets red. Prostaglandins are the type of lipid that regulate, they help with blood clotting, inflammation, the inflammatory response. That's what prostaglandins do, okay? So I want you to know, if I ask you on the exam, what's the biological significance of a steroid? Or what's the biological significance of a sphingomyelin? Okay, I want you to be able to briefly, nothing more complex than what we've written, know what the purposes of these fats. I don't just want you to be obsessed with structures and no structures here. I want you to know the purpose of these biological molecules. So considering what we just talked about, by the way, I want to draw out one really, really, really important molecule. I want to draw out glycerol. Okay, glycerol looks like this. We got 3 carbons, and these top 2 have 2 hydrogens bonded here and here, or sorry, the top and the bottom. And then we got a hydrogen here, and then they have these OH groups. Every carbon gets a OH group. Okay, this is glycerol. This is the backbone for many kinds of fats, specifically triacylglycerols or glycerophospholipids. As I circled on the slide above, they are triesters of glycerol. Okay, and the ester bond is going to connect to each of these carbons via the fatty acids that have carboxylic acid groups. So I want you to look at these structures here from briefly what we've talked about. Could we classify? I know you don't know, you don't have to know these long chains of hydrocarbons. That's not important right now. It will become important. But how would we classify these? Okay, let me help you look at this. So this is what we just drew. This would be glycerol, except for that it's connected to these carboxylic acids. Okay, I'm going to that's carboxylic acid groups, and they're connected. The ester bond is the one that's I've double boxed in the middle. So this has then 3, I'm going to highlight, this has 3 fatty acid chains. These are all going to be fatty acid chains connected to glycerol. So with 3 fatty acid chains, we call these triacyl. This would definitely be a triacyl glycerol. Okay, and what does this molecule look like from the slide above? How would you classify it? Anybody just from looking up here? What does it look most similar to?
Steroid?
Thank you, Maggie. Okay, we don't even know what this molecule is, but we can tell from this kind of these 4 ring structures. That's like the classic structure, kind of nucleus of a steroid structure. Okay, when you see those 4 rings connected. All right, a few more terms to know, little definitions to be aware of when it comes to fatty acids and their esters, okay? So the term fatty acid, I kind of mentioned this up on the first slide. We said they're long unbranched hydrocarbons with a carboxylic acid at one end. Here is a visual of that. Here is our long hydrocarbon tail, and they can be this long or longer. And then I'm going to highlight, here is the carboxylic acid group. Carbonyl carbon with an OH. And get very used to this idea. Yesterday we said this hydrogen right here is acidic. It can come and go. And we're going to see it go a lot because it's going to bond to other molecules, especially when you get triacylglycerols. Okay. Now, many fatty acids are what we call saturated. So they look a whole lot like palmitic acid right here because they contain only carbon-carbon single bonds. Okay, if you look at this structure, all the black spheres are carbons, the white ones are hydrogens, and the red ones are oxygens. So all these black spheres are carbons that you see that they're connected by single bonds. We call that saturated. Unsaturated fatty acids contain 1 or more carbon-carbon double bonds. And in a fatty acid, the dominant form of double bond is cis. Okay, they're usually cis. We prefer cis health-wise over trans. So we have even more specific words though if we have unsaturated fatty acids. Right here we have, if it's monounsaturated, that's the molecule only has one carbon-carbon double bond. Mono indicating there's just one carbon-carbon double bond in the molecule. Polyunsaturated means there's more than one. It could be 2, 3, 4. The number of carbon-carbon double bonds, you will hear it called the degree of unsaturation. Okay, the degree of unsaturation. So for example, look at linolenic acid right here. Linolenic acid, this is right here. This is a cis unsaturated fatty acid. It's got 3 double bonds, and those have the arrows pointing to them. So you can see the molecule is fairly straight, but when you begin to have these cis double bonds, what happens to the molecule? Is it straight? It becomes what we call kinked, like it bends or it turns in different angles. Okay, so cis molecules or fatty acids that have a lot of cis double bonds, they're not straight chains like the unsaturated, or sorry, like the saturated fatty acids. They're twisted and they're turned, and they don't line up perfectly and straight the way saturated fatty acids do. So we also, because fatty acids can be really, really long, and we don't want to draw out the Lewis structure of these, my goodness, that would take forever. We have a shorthand notation to quickly tell us how many double bonds there are and how many carbons there are in a fatty acid. So the shorthand notation, you'll see this on a table down below, looks like this. We draw a C for the number of carbons. We write the number right afterwards. So in linolenic acid, if you were to count up these dark spheres, the black spheres, you count 18 carbons. Then we put a colon and we write the number of double bonds. So for linolenic acid, all these pink arrows, we've got 1, 2, we said 3 double bonds. So then we just write a 3. So if you see this shorthand notation, you see C18, that's the number of carbons, colon, whatever the number comes after the colon is the number of double bonds, okay? And that is what you see right down here on this table. So this is right out of your textbook. And, you know, this is, here's what I'll probably do. You know what? Here's what I decided. Y'all haven't written your note cards yet, have you? Okay. For the exam, I'm going to put this table that you see right here on the slide, that's going to be on a resource page. And then the 5 monosaccharides that I wanted you to know. So like the glucose, fructose, ribose, deoxyribose, I'm going to put those on a resource page so that you can see their structure. I'm not going to put all the extra added info, but I'll at least have a picture of those monosaccharides on the resource page. So don't worry about cramming those on a card. If you want to spend your card writing stuff about the things about reactions that you need to know. Robin, did you share that picture with everybody?
Yes, I sent it in the group me.
Okay, so Robin sent a picture in the group me that has kind of a list of we kind of summarized all the reactions that we've covered. So spend your note card writing things like that, okay? If there's any other tables that I think are critical, I will throw those on a resources page too. So probably by the end of, it might be an email sometime over the weekend when I get it all squared away, but I'll tell you what I'm putting on this resource page. But for sure, this table and the monosaccharides will be on there, okay?
Okay, so this whole table, but just the monosaccharide, like.
Yeah, like I'm not going to put all the extra added.
Yeah, just their forms.
So just the pictures of the monosaccharides probably.
Okay.
Okay, so look at this table. You see the name, whether it's saturated or unsaturated. Typical source you can find it in. It does tell you the number of carbons and double bonds, but here was that condensed notation that I just wrote out for you guys. You're going to use this condensed formula quite a bit. We're going to have some practice problems today where I say write a trans or write a triacylglycerol where 1 unit is oleic acid and 2 units are stearic acid. You're going to come to this table and you will literally grab these condensed formulas to write those out. Okay, do not write the whole chain. That will take forever and a day. Always write them in their condensed formulas. That will help you a lot. All right, and then melting point. Notice this. Look at the saturated fats versus the unsaturated. What do you notice? Anybody notice anything?
The saturated fats are more stable than the unsaturated fats.
Yeah, they all have a lot higher melting points, okay? Coconut oil, butter. I always remember coconut oil because like in the summers, it like is almost melted in my can at home. I always buy big things of coconut oil. And in the winters, it's like always really hard to scoop out. But saturated fats, they have no double bonds. Okay, these have much higher melting points. Some of them very high melting points. Okay, so they're usually solids at room temperature, or they are solids at room temperature. Unsaturated fats, where you start to have 1 or 2 or 3 or more double bonds, these have very, very low melting points. So they are all, do I have space to write? They're all liquid at room temp. And I'll draw an arrow. These are all solids. Okay, so that's a quick way to examine. Like if you sit butter out on the counter, it's going to remain a solid at room temp. Now it'll be very soft, but it'll still be a solid. Okay, but like olive oil, vegetable oil, canola oil, things like that, we know those are, if you buy them, grapeseed oil. I don't know how familiar you are with your oils, but these are all liquids at room temperature. Okay, because of there's structural differences here that are responsible for those properties. Okay. Questions so far? Any questions so far? Okay, now we're going to get a little bit into structure. I actually don't have time to go over the structure of waxes, okay? So I'm not going to ask you to draw the structure of a wax on the exam, but I do, if there is one particular lipid I want you to know how to draw the structure of, that is triacylglycerols, okay? Or we call them triglycerides sometimes. So this is glycerol with 3 fatty acids. So I'm going to zoom in right here just on this like little cartoon structure of it first. The way this works out is we got a glycerol molecule. That's what I call the backbone. And then you have 3 fatty acids all branched off. And the carboxylic acid, I'm going to abbreviate, carboxylic acid group, that is what they are branched through. They're carboxylic acid group. Okay. So if you scroll over in blue, this is glycerol. And it's labeled right down here for you. We drew that molecule earlier. And you can see how they are bonded. I'm going to highlight in yellow the ester bond. Okay, a carbon to an oxygen to a carbon. We know that's a carbon, oxygen, carbon, where one of those carbons is a carbonyl carbon. That's the ester. Okay, just a carbon, oxygen, carbon is an ether. So the fact that we have that one of those carbons is a carbonyl carbon, that's what makes it an ester. All right. Now the R stands for the rest, the long hydrocarbon chain for each fatty acid. And those can all be different. That's why you see R, R prime, and R double prime, okay, with the little quotation marks, indicating that these R groups can all be different. They don't all just have to be R. They don't all have to be the same. They can all 3 be different, or they could all be the same. And that's where you get wildly different triacylglycerols. You can have all different kinds. So for example, we have a one written right below where 1 is palmitic acid, it's saturated, and then you have 2 unsaturated triacylglycerol or fatty acids branched off of glycerol. Okay. So if I show you a structure like this, I would definitely want you to be able to classify, oh, that's a triacylglycerol. I recognize that. Okay. Now I bet you could fill in the blanks over here. Animal fats. Think about animal fats. So if you go to the store and buy a steak, okay, you can usually see the fat marbled into meats, right? This is animal fats. Okay, so they are a larger percent. Do you think they're a larger percent saturated or unsaturated? Are they solids or liquids at room temperature?
Saturated.
Yep. And they're there for solids. Okay. Vegetable oils, so we mentioned this, are mostly unsaturated. And they're therefore liquids at room temperature. Okay. All right. Now, also I want to point out, just because we draw the double bonds like this does not mean these molecules are straight. Okay, remember, anytime there's a double bond, it's cis. And so these 2 unsaturated, oleic and linolenic, these would be kinked. Okay, these wouldn't look nice and straight the way they're drawn. All right, any questions about the structure of triacylglycerols so far? So ignore this top question because that's about waxes. I'm not going to have you do that on the exam, but the next one I do want to draw. I want you to draw the structure of a triacylglycerol. Okay. Now you have glycerol, and then you have 3 oleic acid acyl groups. So this is really good practice. You might have to do this on the exam. Remember, I want you to use the condensed notation or the condensed, right here, the condensed formula. That's what I should say. Okay. So I want you to draw this out. It said 3 oleic acid. So here is oleic acid. Okay. Now you don't have to write the word cis. You can leave that off, but everything else is going to be important. And I want you to notice on oleic acid, that is the carboxyl, that's the carboxylic acid portion. So you can either you can write it this way, where the carboxylic acid part is on the right side, or you can write it where the carboxylic acid is on the left side. It doesn't, or sorry, you could write it where the carboxylic acid is on the left side. Either way, it doesn't matter as long as you orient the rest of the molecule correctly with that. So we've got a CH3, then we've got 7 CH2 groups, if you're looking at this. Then we have a carbon-carbon double bond. Then we have 7 more CH2 groups, and then we have our carboxylic acid. So I'm gonna come down and I'm gonna write this just so that we have it on our sheet before we draw the whole structure of the fat. So oleic acid was CH3, and then we've got 7 CH2 group, 7. Then we got a CH double bond CH. Then we had 7 more, right? CH2, 7 more of those, and then COOH. Okay, this is oleic acid. So I want to draw the structure of a triacylglycerol whose components are glycerol and 3 oleic acid acyl groups. Okay, why do we have this word acyl? Well, when we start to bond things with long carbon portions, sometimes those are referred to as just acyl groups. Those acyl is a very vague term. It can mean a lot of different things. But we're attaching carbon substituents to glycerol here. So I'm going to draw it, since we already have this oriented as is, I'm going to keep it like that. So I'm going to draw glycerol on the right side of my paper. So here's glycerol. You just draw a 3 carbon chain. Now, glycerol is going to be bonded to 3 oleic acids. And it's going to look just like, gonna look just like this, okay? I've just oriented it the other way. I have my glycerol on the right side, but I want to connect glycerol. There's gonna be an oxygen, okay? And then I'm gonna put my carbonyl groups, and then I'm gonna draw the rest of oleic acid, okay? So I'm gonna draw the oxygen here, and then I'm gonna draw my carbonyl groups. Okay, now these carbonyl groups, that's that carbon right there. That's part of oleic acid. Now I need to draw the rest of oleic acid. And I also have to include the hydrogens on glycerol, right? How many hydrogens should I put on each carbon of glycerol? Look at the number of bonds each carbon has. The rest of them that you don't see to get to 4, and how many hydrogens you need. So this first carbon, it's got 2 bonds, so that one needs 2 hydrogens. The middle carbon has 3 bonds, so I just need to give it 1 hydrogen. And the bottom carbon has 2 bonds as well, so I need to give it 2 hydrogens, okay? Now we draw oleic acid. I'm literally just gonna copy and paste this part right here, here, and here. Okay, to finish my triacylglycerol. So I'm going to draw CH2. There's going to be 7 of these. And then I have my carbon-carbon double bond. And then I have another 7 CH2 groups. And I have CH3. Anybody confused on how we wrote this out? If I were to draw this out in line structure form, it would look, do I even have space? I'll draw it up here. CH3, we got 1, 2, 3, 4, 5, 6, 7, and then we would have a double bond. We know it would be cis. And then we'd have 7 more. 1, 2, 3, 4, 5, 6, 7. And then I have they look like they're drawn upwards down below, but the way my lines work out, I'm just going to orient it down. It doesn't matter. We know carbon-carbon single bonds can rotate. and then I would have the O, and then I
Are
would,
we able?
yes, Maggie.
Sorry, are we able to write it the other way? Because
Is it messing
I
you
don't
up that
know.
glycerol's on the right?
yeah, writing it backwards. I feel like I'm dyslexic. I don't. My hand's
Okay,
not.
but if you flip
working
it,
right.
then you're gonna have to literally flip oleic acid and draw it the other way, okay?
So
Okay.
I have a question.
Yes, Robin?
I mean, I'm looking at it and I can see it, but I just kind of want you to tell me so it sticks in my brain.
Yeah.
So on the right, my carbons right here are what is my C bonded to my O, bonded to my O, bonded to my H, right?
Okay, so this yellow part, that is oleic acid,
Yeah, I got that part. So
okay? The blue part, this is my, it's the carboxylic acid, but now it's connected to glycerol, so it's become an ester.
okay.
That's the ester. And then I'll highlight in green, if y'all can see this, this is glycerol.
Okay, so this is glycerol. Oleic acid. Okay, hold on.
Does that help if I do it like that?
Yeah, I'm just like where you put where we got CH2, CH, and CH2. I'm just like how did we get CH2 and then CH and then just CH2? Like, did that come off that COOH? Is what I'm asking.
That's just
Or-
glycerol, this right
Oh.
here. Oh,
Oh, so we just do that every time then.
yes, glycerol is like a backbone. So you're always gonna see glycerol when you have triacyl, when you're drawing triacylglycerols. But remember, triacylglycerols are only one kind of lipid.
Oh, okay. Now I get it. Okay.
Maggie.
What makes it acyl?
Acyl is just a word for a carbon substituent. Okay, so acyl could be you're attaching if we were attaching a methoxy group, so like OCH3, we could call that an acyl group. Okay, don't get hung up on the word acyl. It just happened to be part of 3 oleic acid acyl groups. It just means you have some molecule that's got a carbon portion and we're attaching it as a substituent onto something else. That problem could say, and 3 oleic acid substituents, or 3 oleic acid fatty acid, 3 oleic fatty acids, okay? Or 3 oleic acid groups, okay? They just put acyl, a little descriptive word. Okay, so then we would continue. You would literally draw this part as well. Can I copy it?
Yep.
Boom, like this, okay? So that's what it looked like when you're all done. I know this is new, so I know it's probably not very comfortable, but this is the one type of lipid I want you to be able to draw structures for. And again, I will give you that table. You will not have to memorize what oleic acid is. Okay, properties of fats and oils. You could probably actually, some of this might be intuitive for you now. Since we know fats have such a large hydrophobic portion, like these hydrocarbon chains, it should not surprise us that they are dominantly nonpolar, hydrophobic, and the type of intermolecular force that holds fats together. Like if we look at these 3 chains right here, how are these interacting? It's through London dispersion forces. Okay, it's holding them together. The fact that straight chains can all lay together so nice and neat, the way you see where it says a fat, these are saturated fats, is because you have a high degree, you've got a lot of London dispersion interactions there. This is why they have higher melting points. This is why they're solids at room temperature. It's because of these London dispersion forces. Look at where it says an oil. This has got to have some sort of cis double bonds, right? So all the fats are all, it looks like, strings of spaghetti in all different directions. We've got space here, so we don't have London dispersion interactions all down the chain the way you see in saturated fatty acids, okay? But they are nonpolar, so the dominant type of intermolecular force is London dispersion forces. No ionic charges. So solid triacylglycerols, fats, high proportion of saturated fatty acid chains. We've already hit on that. Liquid, high proportion of unsaturated. Okay, I feel like we hit that like 3 times now. This is just showing you the difference between saturated and unsaturated and what the cis double bonds do and how they kink the molecule and turn it. I definitely on the exam want you to be able to relate melting points, if something is a solid or a liquid at room temperature, to the degree of saturation or unsaturation. Okay. This is just a reminder, the more double bonds you have, the lower the melting point, because the more kinked and spread out the fatty acids are and the less intermolecular interactions, London spread forces you have between those fatty acid groups. Okay. This is really important. I want you to know the primary function of triacylglycerols. Long-term storage, we said, of energy. We wrote that on page 1. But what else do they do? Okay. Adipocytes, you probably heard that. This is fat cells, fat tissue. Adipose tissue provides thermal insulation to our bodies and protective padding. Okay. So you should be thankful for your fat, for these things it does for you. All right. This is also not going to be brand new information. When we talked about alkenes and alkynes, we actually talked about hydrogenation. So I introduced this word back on day 2. So chemical reactions of triacylglycerols, you can take unsaturated fatty acids and hydrogenate them. Okay, this is done commercially. So it's what we do is hydrogenation is the addition of hydrogen to the double bond of an unsaturated fatty acid to yield a saturated fatty acid. We can synthetically turn unsaturated fatty acids to saturated fatty acids by adding hydrogen across the double bond. So you are familiar with this kind of reaction. We're just now applying it to fatty acid molecules. Okay, so what happens is the carbon-carbon double bonds are reduced stepwise, not all at once, one at a time. So they're reduced in a stepwise fashion, one at a time. So the degree of hydrogenation, in other words, the number of double bonds you convert to carbon-carbon single bonds, can be regulated. Okay, so industrially, they could stop it at any point in time once they feel like they've got the type of fat that they want. Okay. The risk here is that hydrogenation can result in our trans fats. That's how we make trans fats. And we said these are associated with different post-health risks because trans fats don't occur naturally. They're created synthetically. Okay. So before you go deep fry that turkey for Thanksgiving, although let me tell you, I'll be the first to say, deep fried turkey, anybody ever had it? It is so good. It's so good. Okay. But Cassie's not going to have that for Thanksgiving. She's going to have tzatziki's. Okay. But I would not be mad if I ate tzatziki's on Thanksgiving. It's that good. Okay. So this is just showing you an example where we're adding hydrogen across these double bonds. Okay. And you end up with the carbon-carbon single bonds, and we call those hydrogenated, or we call the process hydrogenation. Okay. Questions on this? Okay. Hydrolysis. Y'all should be super familiar with the word hydrolysis by now. But don't read the slide. Somebody tell me, what is hydrolysis? What do we do? What does hydrolysis do? For any molecule, not just fats. It's the use of water to do what? It's the use of water to, what are we doing in hydrolysis? We're breaking apart molecules. Okay, we're using water to break apart molecules. So in hydrolysis of fatty acids, same idea here. It's the reaction of triacylglycerols with water. We're going to break it apart into its original parts. Okay, carboxylic acid and alcohols. Okay. Now we have enzymes that do this for us. Okay, they're called hydrolase enzymes. Notice the ASE ending indicates an enzyme. This right here, ASE hydrolase, tells us it's an enzyme. Catalyzes reaction. When we have to digest fats, we have to break fats apart. So we have enzymes that do this for us inherently. Commercially, okay, in the lab, this is done with really strong base, such as sodium hydroxide. And commercially, we call the process saponification. The word is also at the bottom of the slide down here, saponification. This is how we get soap. This is how we make soap, by the way. We know soap and water doesn't mix, right? So water's polar, soaps are non-polar. Soaps are fatty acids. So the way we do this, the way this works, saponification, here is glycerol. Okay, y'all recognize glycerol. There's glycerol. So orient yourselves here. Here's the ester portion. And then R, that's the fatty acid. Okay. This would be the long hydrocarbon chains. We're just not drawing out the long chains. All right, so when you react this with NaOH, here's what happens. I'm gonna draw the bond that breaks. We're gonna break glycerol apart from its fatty acid, and we're gonna break it right here. We're gonna break these bonds between our glycerol carbons and the oxygen that's part of that makes the molecule an ester. Okay, we're gonna break those oxygens away from glycerol. And the OH, so I'm gonna highlight this in yellow because the OH portion of NaOH, that is gonna bond to, it's gonna remake the glycerol molecule. So the OH from NaOH, the strong NaOH is a strong base. That's gonna bond to restore a glycerol molecule, and then the sodium part, I'll highlight that in blue because that is going to be attracted to the negatively charged carboxylate. Remember, when you have a carboxylic acid and it gives away its acidic hydrogen, what you're left with, we call this part now, that it has, see the negative charge on it? We call that. These are carboxylate anions. So when we break it apart from glycerol, we end up with carboxylate. These are fatty acids that have a carboxylate anion at the end. And that negative charge is attracted to the sodium, the positive charge. And we call these, so you can read down at the bottom, we call these fatty acid salts because you've got positive, negative charge here. It's an ionic compound in a sense. This is soap, or a form of soap. Okay, question on how saponification works. Now, how is this useful biologically? What's the significance here? So these fatty acid salts or soaps, they work in spherical clusters called micelles. Anybody heard the word micelle before? And a micelle, once we mix it with water, okay, there's a part to the fatty acid soap that is water loving, and that's the charged parts, the carboxylate anion part or the cation part. And then there's a part that's water fearing, and that's these long hydrocarbon tails. So these long hydrocarbon tails right here, these are water fearing. These are hydrophobic. And the purple heads or spheres, these are water loving. These are hydrophilic. So they will arrange themselves into what we call micelles, and a picture of it is right here on the right. They'll form these spheres where all the water loving heads are on the outside, the hydrophilic heads, and the hydrophobic tails are on the inside. And this is where it traps dirt, grease. This is what, when we wash our hands with soap, this process is literally happening. When we wash our face with face washes, it gets the oils and the things off. Okay, makeups, cosmetics. It's this process right here, micelles. Okay,
Oh, is that why micellar water is called that?
it's why you're what?
Do you ever use micellar water to take off your makeup?
No, but I bet that's what it is. Why it's called that. You're trying to be extra sciencey.
That's funny.
Okay. So we're gonna practice this one time. We're gonna write the complete equation for the hydrolysis of a triacylglycerol, in which the fatty acids are 2 molecules of stearic acid and 1 molecule of oleic acid. And I have put the condensed formulas for oleic and stearic acid right here. And for Maggie, this time I'm gonna write it the opposite way. Just for Maggie, because it's her birthday week. Okay, Cameron, I guess you too.
Can you
All
write
right.
it in the opposite way and then one the?
So we're gonna write glycerol on this side. I just want y'all, and I want y'all to not be freaked out no matter which way you write it. Oh, that should just be H, not H2. Okay, so this is glycerol. And we're gonna draw it to, we draw the ester bond right here and then draw the carbonyl carbons. And now you begin your formulas for the fatty acids. So for stearic acid, we'll put stearic acid first. So now we have to take this right here and we have to flip it the other way, because this carbon right here is that carbon right there. That's our carboxylic acid carbon. And we've already drawn the oxygens. So now we just say, okay, we're gonna have for stearic acid, CH2, we're gonna have 16 of these, and then CH3. Stearic acid's nice. I like stearic acid. It's very simple to write. The other two are going to be, 2 molecules of stearic acid, sorry, 1 molecule of oleic acid. So let's write the middle one is stearic acid too. CH2, 16. Maggie, do you like this better?
I'm sorry, I was writing it. What'd you say?
So do you like this better?
Kinda.
Okay. So there is our triacylglycerol. Now we're going to hydrolyze it. Okay, y'all got to listen faster because we have a lot of slides still to cover. Okay, so we're going to break these bonds. And now we draw our products. So we're going to have glycerol. And I don't care how you write glycerol. You can write it like this, or you could write it like this. Either way. Okay, there's glycerol. And we're doing hydrolysis. So we're breaking apart. I forgot to write in the water. We're breaking this apart with water. So what's happening is we got H and we got OH. The OH part is what's going to bond over here to the glycerol carbons, all 3 of them. And in red, I'll say the hydrogen part, that is what's going to come over here and bond to that oxygen on each fatty acid. So now, for time's sake, here's what I'm going to do. Don't do this on the exam. I won't give you points, but I'll just say LARI, stearic. Whoops. We will get, I don't need to connect them. We get stearic acid again. And then we get oleic acid. Okay. Okay, anybody confused what this is hydrolysis? You guys see how this is hydrolysis? Well, so these are the red hydrogens from water. These are the blue OH groups from water. We've used water to break apart and to hydrolyze our fatty acid.
Do we need to put a plus sign in between the two that were broke apart?
Sure,
Or like with that?
that's fine. Okay. If I gave you a fatty acid and I said, write the products from hydrolysis, could you do it? Yeah, you could. All right. You know what I'm gonna do? I'm gonna jump over a few slides for time's sake, because we really need to get to cell membrane transport. So if I don't cover something in this lecture, it will not be directly on the exam Monday. I'm gonna try to come back to this, but I bet time's not gonna allow. I do want to point this out. Sphingolipids, we talked about those. Right here. We said they're the major components of nerve fiber coatings. Okay, they protect our nerve cells. I wanted to point this out because deterioration of sphingomyelins is associated with multiple sclerosis. Okay, so a very common way they diagnose multiple sclerosis is you can literally see it on like CT scans. They'll take like a spinal scan and a brain scan, and you'll see where like there will just be like black spots at the base of the brain or along the spine that show deterioration of this is why it's called the myelin sheath, right? Because sphingomyelins, okay? These are the fats that coat our nerve fibers, and they're really important for that. Okay, let me keep going. Real quick, I'm going to point this out. So glycolipids, we said the major importance with those. I don't need you to know the structure, but we said these are intracellular messengers, and they stick out on the surface of the cell membrane, okay? And there's two different kinds. You've got cerebrosides and gangliosides, okay? So if you look over here, these glycolipids, I'm going to point one thing out on them. The reason why it says sugar side chain is because on part of the fat, part of the lipid molecule has literal monosaccharides. Okay, look, there's galactose. We saw galactose yesterday. So it's like a fat that has sometimes a sugar polysaccharide or monosaccharides attached to the fat somewhere. I'm not going to have you draw this, but if it's a cerebroside, then the carb portion that's attached to the fat is just a single monosaccharide. Okay, so I imagine it might look something like that. Maybe that's a cerebroside. You just see one little thing poking out above this is a lipid bilayer. If it's a ganglioside, then the carb is a small polysaccharide, and that's these green right here, okay? They stick out above the cell membrane, and they act as receptors for cell-cell messaging. Our cells are communicating all the time, and they're receptors for cell messengers. They communicate inside the cell. So those would be gangliosides and cerebrosides. Okay, these are types of glycolipids. All right, and that's essentially what these two bullet points are, what I just said. Okay, they're found within cell membranes, but they extend out into the extracellular environment. We're skipping over this. Don't worry about this. We're skipping over that too, because we just don't have time to do it. All right, let's get to sterols. So again, we skipped over a lot. So what's important to know, what's not important to know? I just want you to know the significance of the types of lipids we skipped over. The glycerophospholipids, the sphingomyelins, and the glycolipids, okay? Know what they're used for, what the function of those lipids are in the cell and the body. Sterols, okay? So these are lipids whose structure is based on a fused 4-ring, so we will say tetracyclic carbon skeleton, okay? So this is the carbon tetracyclic skeleton of steroids. They can have all different kinds of groups attached, branched off, but you'll commonly see this 4-ring structure, okay? So again, they're not soluble in water, hydrophobic, that's classic for all lipids. Major sterile function. So cholesterol is the main sterol in the human biochemistry, but you also have bile acids. So your gallbladder is where we store bile acids. And these are essential for digestion of fats and oils. Okay, we have to break down fats. We have to have these bile acids kind of act as emulsifying agents. Does anybody know what the word emulsifying means?
Isn't it kind of like fusing like together?
Yeah, emulsifying agents will have like both polar and non-polar portions, okay? Because you're trying to, once you eat and ingest a fat, your body is aqueous. It's got a polar environment, okay? Our blood has got all sorts of electrolytes in it, water content. So we are trying to mix these fats with an aqueous polar environment. So emulsifying agents help do that, and they really, really help in digestion with breaking apart and our fats and mixing them with the rest of our body, okay? Anybody ever heard of a gallbladder cleanse before? So I went to a chiropractor back in the day, and I just remember him talking about a gallbladder cleanse. It was the most disgusting thing I've ever heard of. Basically, you like eat apples and pectin for like 3 days straight. And then he's like, stay home for 3 days straight because cleansing your anyways, it's a very bizarre thing, but bile acids make me think of that. And then you got hormones, okay? So these are all different types of sterols. So we said cholesterol is the most abundant one. What's cholesterol used for? Well, it's part of our cell membranes, okay? And it just kind of helps hold them together because fats can be, they can have these long hydrocarbon chains. They can be very wiggly, depending on how many unsaturated bonds you have in there. But cholesterol kind of helps like solidify the cell membrane and keep it together. So it increases rigidity. And it's also the starting material for the synthesis. We make a lot of our own things internally too, okay, in-house. Some things we have to eat through the diet, but our body also makes a lot of the essential things we need. And so we synthesize other sterols, and cholesterol is the molecule we use to make other sterols, okay? So there is, I didn't label this, but I believe that's cholesterol right there. Okay, so a little bit more on bile acids and steroid hormones, and then I think we're really on good track to talk about cell membrane structure and transport.
Oh, question real quick.
Yes, Robin.
Did anybody else's PowerPoint stop after sterols?
Yeah, I was about to say I only have up until sterols. It was only
Really?
19 slides.
So y'all don't have cell membrane structure and transport?
I was wondering why it was so short today. Like,
Right, you was rushing. I was like, is
Yeah,
this one
same.
slide gonna take 30 minutes?
Weird. Okay, let me email this to y'all real quick. I know you won't have it printed out, but at least you can look at it on your screens.
Did you update it on Blackboard?
That's bizarre. Maybe I just uploaded the wrong one to Blackboard.
Here, well, let me see if it's updated on Blackboard real quick. I can see.
I didn't. I don't think I did. If
I printed it off at the beginning of class and I don't have it either.
right, so
Oh,
I
okay,
put the wrong
that's
one on
fine.
Blackboard. Is probably what I did. Okay, Maggie and Madison and Abby and Cameron and Robin. Okay.
How much more of the cell membrane structure and transport slides do we have? Like, can you upload it real quick?
That's
if
what
you
I'm
don't-
emailing it to you guys real quick.
Oh, okay, okay.
Yeah, I'm just attaching it right now.
Oh, dang.
Chapter 23, here it is right here. Which one's a PDF? Where is there? No. I just saved it as a PDF. Well, so what's great, I guess I do a lot of this late at night, which is probably why there's a mistake here. Yeah, chapter 3 Lipids PDF. So I need. There it is. All right. Open and send.
I mean, we could always just stop at this
Oh,
sterile.
no, Robin. I expected nothing less. Okay. I just emailed it to you guys. So check your emails. Do y'all see it? And this should have, I mean, this is what I literally just uploaded to my iPad. So yeah, I see all the slides at the end there. So there's 1, 2, 3, 4, 5 slides, and then there's a practice slide.
Anybody got it yet? No,
I haven't gotten it yet.
me either.
I just got it.
Just got it?
I just got it.
Sorry about that. If you have to print this, just print the last 6 slides. I think it starts with slide number. On my PowerPoint, it is slide number 20 through 25. I don't know if that helps. Okay, everybody got it now? Who's still waiting on it? Okay, sorry. Where were we? On sterols. So I'm gonna briefly finish up sterols, bile acids, and steroid hormones. And just kind of the purpose of those and some common ones. So we said bile acids are emulsifying agents. So they emulsify fats, they help break down fats during digestion. Okay, and this is happening in your intestines, right? Not your stomach. This is once food goes through the stomach, gets into the small intestine, where we really start breaking it down. Bile acids are stored in the gallbladder. And how do we know, how does our body know when to release them, when to not release them? Well, anytime we eat a meal, there's all kinds of signals going off through our body that lets us know. So meal consumption is what stimulates their release into the small intestine. This should not surprise you, largely hydrophilic, because they have to mix. They have to be both hydrophilic and hydrophobic. So the ring, so let's look at, here's 2, cholic acid and chenodeoxycholic acid. I probably didn't say that right. But you see all these OH groups, okay? And you see this carboxylic acid portion here. And so we're starting to see some more polar groups on things like bile acids. They're still classified as lipids, but these polar regions help the molecule function as an emulsifying agent, help aid in digestion. I wanted you to look at this, can you circle the hydrogen that would be lost to yield the anion form? Because we said anything that has a carboxylic acid on it actually exists as the carboxylate anion at physiological pH. So I circled 4 OH groups there. Which one is the hydrogen that's lost? Which one's the acidic hydrogen?
It's the top one, right?
It would be the one I'm going to highlight right here. That's the acidic hydrogen. That's the acidic hydrogen. Okay. And that's what this last bullet point says. They exist in their anion form, and that's why they're called bile salts. Bile acids, bile salts. All right, so that's so we got cholesterol, bile acids. Last one, steroid hormones are a type of sterols. So I just want you to know these 3 steroid hormones, mineral corticoids. So they regulate sodium, potassium levels in cellular fluid. Sodium and potassium are 2 really critical electrolytes in our body one for our nerve function. And then we maintain various gradients, concentrations of these electrodes across cell membranes for many reasons. We're going to get to gradients in just a minute. So mineralocorticoids, those are a type of steroid, and that's what they do. Glucocorticoids, hydrocortisone. Okay, I have hydrocortisone at my house, right? That's an anti-inflammatory. Anytime you get like a bug bite, or you might rub hydrocortisone on it. This is also involved in glucose metabolism, glucocorticoids, and then you have sex hormones. So androgens, that's like these are responsible for, what are these responsible? These are responsible for male secondary traits. Okay, biological. So what would be like a male secondary trait? Okay, males typically have Adam's apples, deeper voices, things like that. Those are male secondary traits. And then you have estrogens that are responsible for female secondary traits. Okay, and so there's many things, enlarged breasts, pubic hair, different things that estrogens are responsible for on females. Okay, and then androgens for males. So here's a picture of testosterone. You're not going to have to draw these out, okay? But if I gave you a picture of testosterone, or I ask you what they're responsible for, I want you to be able to tell me. All right. Cell membrane structure and transport. So these last 5 slides, I think we're going to have time to do them. So we cannot not talk about the cell membrane. Okay, when we talk about lipids, because lipids are the primary component, not the only component, but the primary component of our cell membranes. And our cell membranes are critical for so much, okay? We have to have this protective coating around our cells for so many reasons. So really quick, let's look at the structure of a cell membrane, and then we're going to talk about how it functions and how it operates. So they are composed of phospholipids. These are the basic structure of the bilayer. They are ordered and stable, but they're also flexible. Okay, so that sounds a little bit contradictory. There is order to our cell membrane. Okay, you can see the order in the structure right here. But things can move through it. Okay, they can allow molecules in and out. They have to. The reason it puts itself in a bilayer, we call this the lipid bilayer. So there's 2 layers, and that's because we have every fat or every lipid has a polar head that is hydrophilic, and it mixes very well with the extracellular aqueous environment. But then it has the nonpolar tail that is very hydrophobic, water-fearing. So they orient themselves this way to put like portions together. Those nonpolar tails, and then those hydrophilic heads can interact with both the cellular internal membrane, which is internal environment, and the extracellular environment. Those are both aqueous. Okay. Another way they can orient themselves, though, is in what we call a liposome. And I want you to know, how are liposomes and micelles different? Here's how they're different. Liposomes have a bilayer. Micelles don't. Okay, liposomes have a bilayer. Micelles don't. So in the very center of a liposome, I'm going to draw the arrow here. This is aqueous. When I say it's aqueous, I mean it's just like the internal cellular or extracellular environment. You can put polar things, like polar molecules. You could put polar drug molecules in the center of liposomes. In the center of a micelle, the environment is entirely nonpolar. Okay, that's where the grease and the dirt and the other nonpolar things go. Okay, you don't have this central polar environment in a micelle the way you do for a liposome. Okay. So liposomes can store water-soluble substances on the interior. They are useful for drug delivery. That's one function of them. Okay, what else is in our cell membrane structure? Glycoproteins and cholesterol. We already talked about what glycoproteins are for. They have those sugar polysaccharides or monosaccharides that stick out above the cell surface and act as receptors. They're important for intracellular messaging. Cholesterol, we said, is important for the rigidity of our cell membrane. So those are part of it. And then we have proteins. Proteins compose about 20% of the mass of our membranes. Okay. Now we have different kinds of proteins in our cell membrane. We have some that go all the way through the membrane and some that don't go all the way through. So we have integral proteins. These do extend fully across the bilayer. Why? Why would we have proteins in our cell membrane? Because they act as channels for the transport of various molecules and ions, specifically molecules and ions that might be polar or ionic in nature. Okay, consider the bilayer. Again, I'm going to scroll over here to this picture. We have this non-polar internal region. Ions and polar molecules do not want to pass through that. They don't mix, they don't like each other. But when you have something, okay, that is like a channel or a pathway through the membrane, that can facilitate passage of these ions and polar molecules. That's essential because we need all kinds of ions and polar molecules in our cells. So because we have all different kinds of molecules, it's just such an eclectic mix of molecules. We call this, you may have heard this before. We describe our bilayer and our membranes as a fluid mosaic model. Okay, this is what represents the overall structure of our cell membrane. Fluid, why? Because the cell membrane isn't rigid. Things can pass through it, okay? It's fluid-like. But mosaic, because all different kinds of molecules compose our cell membranes, okay? So fluid mosaic model. So this is a really great picture, right from your textbook. And it shows all different kinds of things. Okay, it shows, number one, I'm going to zoom in. You can see the fats, the fatty acids, the hydrophilic heads and the hydrophobic tails. All right, and those are pointed out for you. What else can we see? Okay, we can see molecules like cholesterol. Okay, it's just an example, all throughout the cell membrane, helping to hold it together, helping to keep its shape. Okay, you can see these are called glycoproteins. They can look all different kinds of ways, but what do we notice on them? We notice these sugar polysaccharide units. And these are important for cell messaging. They act as receptors. We mentioned that. All right. You can see proteins. Okay, so there's two different kinds of proteins over here. Integral protein that extends all the way across the channel. But I didn't mention this, the other kind of proteins, the ones that don't extend across the membrane, but might be attached to or just inserted within the membrane, we call these peripheral proteins, okay? So integral extends all the way through the membrane, and peripheral is just attached to or part of, but it doesn't go all the way through. And these are important for various reasons. We're really not going to get heavily into peripheral proteins, but they just help integral proteins operate a lot of times.
Are we gonna need to remember like this picture, I guess?
That would be a very good thing. I think this helps clearly, it's a good visual for you to see how things fit together and remind you of all the functions of things. I would definitely know this picture. All right, now I want to point this last thing out. Some integral proteins are straight channels. So maybe white would be the best color for this. There's a pathway all the way through. Some are gated, okay? You can get here, but then you pause, okay? It's like when you're trying to get into a theme park and you have those like little gates, you have to stick your ticket in and then you can walk through, okay? Some are gated and you have to have a conformational shape change in the molecule or in the protein before the gate opens and the molecule can pass through, okay? Okay, just like at a theme park, you want to make sure who you let in is there for the right reasons. Or even you can think of the door to your house, right? So it's just going to check to make sure the molecule is actually a molecule that needs to be in the cell before it lets it through. It's just a type of way to facilitate and regulate molecular flow in and out of the cell. So some are gated, some are not. Okay. All right. Now, I want to ask you this question. We're about to talk about how molecules are transported into and out of the cell. And there's different ways this happens. But why? Why would it be important for a cell to regulate how different kinds of molecules are transported across the membrane? Anybody think of why this would be important?
I know for the potassium sodium pump, you want to make sure that we're getting enough sodium and potassium that goes through, because you won't want too much of either.
Right? We have to maintain those particular ions at very specific concentrations. So we have to regulate how much of each ion goes across. That's a good one. Anybody else think of a reason? Okay, so with what Madison said, let's jot that down. One reason is because we have to maintain, so I'll say specific concentrations of molecules or ions maintained, okay? Kind of the word that summarizes this is why we have gradients. This is what we call a gradient. When you have various concentrations on the inside of or the outside of the cell, we say that is maintained at a gradient, okay? What would be another reason that we have to regulate? So the reason we have to use channel proteins or gated proteins. Well, some molecules simply won't pass through the bilayer on their own. Some ions will. Look at this, like this guy. It looks like he can just go freely back and forth across the membrane with the help of no proteins. Some ions, some small molecules can do that depending on their hydrophobic or hydrophilic nature. But some molecules aren't going to want to pass through the membrane. What kind of molecules would not want to pass through the membrane on their own? Any ideas?
Would hydrophobic ones not want to go through?
Hydrophilic ones.
Hydrophilic, yeah. Sorry, I got this mixed up.
You're good. I do that all the time. They simply won't pass through the membrane on their own. Okay. There's another kind of molecule I could think of that won't pass through the membrane on its own. Really, really big molecules like proteins. So that's why the cell makes proteins on the inside of the cell a lot. But some molecules are not going to pass through the membrane on their own, and especially depending on size. Okay, so big need help getting through. So based on size, we have to regulate them. We have to help them across. Any other reasons y'all can think of? That's pretty good for now, but I like this picture because it gives you an overall summary of everything we're about to talk about. The different types of transport that help regulate the flow of molecules, all different kinds of molecules in and out of the cell. So we're going to talk about simple diffusion, facilitated diffusion. These are both two different kinds of what we call passive transport. And then we're going to talk about active transport. So overall, you have two different kinds of transport, passive and active. And it's just that with passive transport, you have two different kinds of passive transport. Simple diffusion, things can just flow freely on their own, or facilitated. They can flow freely, but they have to flow through something. Okay. So there's all different kinds. You can see simple diffusion. Molecules can pass straight across the membrane with no help of any kind of protein. Other molecules, they can flow freely across the membrane, but they just pass through a protein channel because you know what? They might be a little bit hydrophilic in nature and they just don't want to mix well with the inside of the membrane. Okay, then we have facilitated diffusion. They'll pass through an integral membrane, but it might be a gated protein. Might be or sorry, I said integral membrane. I meant an integral protein, but it might be a gated protein. Maybe they pass halfway through and then some kind of shape change has to occur and before they can pass the rest of the way through. So that's facilitated diffusion. And then some of them require energy. Okay, that's active transport. Some of them won't get through. That'd be like a big molecule coming through. It just won't get all the way through unless we apply some energy to help it through. Okay, and that's active transport. Okay. Passive transport does not need energy. Active transport does need energy. So what kinds of molecules might we see using each kind of transport? Okay. Actually, I'm just going to highlight some of this. So passive transport, simple diffusion, this is the easiest kind. It's just the normal movement of molecules along a gradient. Notice that, along a gradient from areas of high concentration to areas of low concentration. I might drill you on gradients. Here's why. You need to understand, I would always ask my 1402 class when we get to like osmosis, why gradients are important, how molecules move along a gradient. I want you to know they move from areas of high concentration to areas of low concentration. So the cell maintains two different areas on the inside and the outside. And they move from areas of high concentration to areas of low concentration. Okay. CO2 waste, O2 nutrients, steroid hormones. Okay. So think about this. CO2 and waste this accumulates on the inside of cells. So all of a sudden, as cells operate, we have a gradient. We have a large concentration of CO2 and waste on the inside of the cell. Okay, just like our houses, we accumulate trash on the inside. We put it in the trash can, clean up our bedrooms, our kitchens, or whatever. Put it in the trash can, and it accumulates in our trash cans, in our bathrooms, in our bedrooms, and then we what? We take it outside. Okay, and then it goes. Same idea with the cell. O2 and nutrients. The cell needs these things. The cell doesn't make its own oxygen. So we breathe it in, and then our blood is responsible for taking it. It's highly concentrated in our blood, oxygen and nutrients. This says, so we have a high concentration of O2 in the blood, and we have a low, I'll draw an up arrow, high concentration in the blood, low concentration of O2 in the cells. That sets up a gradient. That is why oxygen moves from our blood to our cells, because of the gradient. I'm going to go to the next bullet point. Small hydrophilic molecules. Hydrophilic, water-fearing. Okay, so that means they'll mix with the inside of the bilayer just fine. Those will be transported through the integral or internal aqueous environment of integral. Sorry, I got it backwards. Small hydrophilic, so it means they're water loving, so they have to pass through a protein. Okay, they are not going to mix with the hydrophobic internal portion of the bilayer. So what would be an example of a small hydrophilic molecule? A water loving, a polar molecule. CO2 and O2, right? Now, overall, if you were to see me in 1402, the previous chemistry class, I would tell you this molecule is nonpolar because the dipoles cancel, and they do. But because we have these lone pairs that are very electronegative, and because we have polar electronegative atoms, this molecule has to pass through integral proteins. Same with oxygen. Facilitated diffusion, passive transport. So these molecules bind to integral membrane proteins, and then the protein changes shape so that the molecule is released on the other side of the membrane. Glucose is a great example of this. Okay, glucose is C6H12O6. We drew glucose a whole lot yesterday, you know. We drew it like this. And then it's got like, I don't remember which way the OHs go off the top of my head. Okay, but we know we got all these OHs. And for sure, one of those should be up or down. Okay, so we got all these OHs. So this is a very polar molecule. And it's a little bit bigger than carbon dioxide or oxygen. So it's going to need the help of a protein channel as well to pass through cell membrane. And then I'll tell you this, once we get it on the inside of the cell, immediately the first thing we do is we phosphorylate it. We'll add like this PO3 group. Why would we phosphorylate it? Why would we add this group, this phosphorus that has all these oxygens on it that sometimes carry negative charges? That keeps it on the inside of the cell membrane. It keeps it from going just right back out. Okay, once you phosphorylate it, it's stuck on the inside of the cell. And then the cell can use it as it needs to. So that it stays in the cell. Okay. All right. So that's passive transport, simple diffusion, and facilitated diffusion. Then active transport. So active transport, again, concentrations of certain solutes must be maintained as gradients. So there are times where we are going to push molecules across the cell in a direction that they may not want to go. Okay. So maintaining concentration gradients requires energy and the movement of molecules through what we call a protein pump. Okay, and this is what Madison said. The example was with sodium and potassium. So we have a specific pump in our cell membranes. It's called the sodium potassium pump. So to maintain concentration of sodium potassium across the cell membrane, we have to constantly be pushing those molecules different directions. Okay, so you will see the sodium potassium pump. It's called sodium potassium ATPase right here. It is constantly grabbing potassium from the extracellular fluid and bringing it into the cell. Okay, and it's constantly grabbing sodium from the inside of the cell and pushing it outside. Okay, so you see these arrows. We are constantly pushing sodium outside and we are constantly pulling potassium inside. So we want to maintain high concentrations of sodium outside the cell, and we want to maintain high concentrations of potassium inside the cell. Okay, so that these molecules on their own might flow the other directions down their gradients from an area of high concentration to low concentration. But we constantly have to maintain the gradient using these protein pumps. Okay, that requires energy to push. So we are pushing these molecules against their gradient. Gradient means what potassium really wants to do is it wants to flow this way from an area of high concentration. So this would be high potassium concentration. This would be low potassium concentration. That's the way it wants to flow down the gradient. And it will do that, but we have to maintain the gradient for it to do that. And that's what the pumps are used for. So it's constantly, pumps are constantly pulling molecules or ions against their gradient. Does that make sense? It requires energy to do that. All right, little bit of review, and then I think we're done. So cell membranes are composed of fluid-like phospholipid bilayer. Small non-polar molecules are transported by simple diffusion, okay? Simple diffusion, passive transport, but specifically simple. Whoops. Directly through the lipid bilayer. Ions and small polar molecules are transported by simple diffusion through protein pores. Okay, these are not pumps. They're just protein channels, or you could call them channels. Protein pores or channels. Glucose, another example of a molecule that would use facilitated diffusion would be amino acids. So glucose and amino acids are transported via facilitated diffusion, okay? So they're going to use protein channels that might be gated, okay? But they're still not requiring the energy, still not active transport. Substances maintained in concentration gradients require protein pumps and energy for active transport. Okay. So if I ask you on the exam, this is a great practice problem based on what we just talked about, the transport. Oh, I actually answered this for us. Probably didn't mean to do that. Okay, so what do y'all think about this first one? Does an NO molecule cross a lipid bilayer by simple diffusion? So again, this could be, it flows directly across the membrane, no help, or maybe it flows very easily just through a protein channel or pore, still with no energy. It's not a gated protein. What do y'all think? Is it a small enough molecule? Even if it's small and hydrophilic, it can still pass using simple diffusion. What do y'all think about that? Think about nitrogen and oxygen. What kind of atoms are these?
Are they polar?
Yeah, these are electronegative atoms. Okay, they're close to fluorine on the periodic table. And because of that, this is a small polar molecule, which means it's hydrophilic. So it's small, hydrophilic. So what do we say? Passive transport, simple diffusion. That is exactly how small hydrophilic molecules are transported. Now they might have to pass through an integral protein, but it happens very simply through simple diffusion. So NO is small enough it is hydrophilic. So it may use an integral, protein. I actually already answered this next question. Okay, the first step in glycolysis, this should be a T right here. The first step in glycolysis is the phosphorylation of glucose to glucose 6-phosphate. Why does this step prevent passage or passive diffusion of glucose back out of the cell? And I drew out a phosphate molecule, or it's not the complete, but you see the electronegative atoms, you see the charges on the oxygen. So this makes glucose a lot more polar. We add this really charged polar group onto glucose, and that prevents it from just flowing right back out of the cell through the membrane. Okay, the last question, the composition of the inner and outer surfaces of the lipid bilayer are different. Why do these differences exist, and how might they be of use to a living cell. I'm gonna let you think about that one. We're at 10:40, we're kind of at time. Answer that one. Spend some time thinking about that one. I really like that question. Okay, so obviously the outer surface is hydrophilic, the inner surface is hydrophobic. Why do we have this difference? How are they used for the cell? So I'm gonna let you think on that one on your own. I think that puts us at time for the day. Actually, 10 minutes passed, but we got through it. Are you proud of us? We got through a whole lecture in the amount of time we needed to. We don't have to come back for an hour. So that was a lot. And if I can clarify anything for you guys, please let me know. There is one question from lipids, but it's not like a hair splitting detailed one on the quiz. Okay. So if you're paying attention at all, that question should be easy for you. And definitely look at this picture, study this picture, be able to label or draw things on a cell membrane if you needed to. Know why we say it's a fluid mosaic model. And if I pointed something out as structurally important, I think the two places I did that the most were with sterols and with triacylglycerols. Okay. Those were things where we actually practiced drawing the structure of triacylglycerols, and then I really pointed out the basic skeletal structure of sterols.